First, there was nothing. And then there were gametophytes, and shortly thereafter sporophytes. But researchers put an end to that because they wanted to study a gametophyte-only universe. These gametophytes could reproduce asexually using spores. The spores could travel through the air and create new haploid gametophytic bodies. The gametophytes could only reproduce sexually when it was wet. Sperm would swim from one plant's antheridium to another plant's archegonium and create a new sporophytic, spore-producing body. To better utilize their environment's nutrients, the sporophytes began growing roots and simple vascular tissue. To prevent water loss, they began growing waxy cuticles, which required stomata to allow air and CO2 into the cells. With extra water and nutrients, some of the gametophytes could grow taller out of the boundary layer, which presented a problem to sperm, which could no longer swim from one plant to another. A solution for some was to become hardier so they could be driven from plant to plant by splashing water droplets. Pollination droplets on the antheridium of plants made it easier for sperm to stick. But the true solution came in a sporopollenin coating and elaters, which allowed the plant to fly through the air despite dry conditions and low viscosity to create new sporophytes. Microphyls soon gave way to more efficient megaphyll leaves on plants. As they competed for nutrients, they developed deeper root systems. Some even grew taproots and developed relationships with mycorrhizal fungi to better collect nutrients and water. Other plants began relationships with prokaryotes capable of fixing nitrogen in cysts in their roots. More resources allowed the gametophytes to grow taller still. Xylem and phloem were initiated to supply the leaves with water and the roots with sugar to allow growth. However, these new, highly vasculated adult bodies were less resistant to desiccation than their ancestors had been. One solution was to reproduce through root offshoots, so that meristems could be stored underground. Here, they could survive to grow grass-like structures even during conditions of drought. Because the gametophytes had haploid adult bodies, they were particularly vulnerable to somatic mutations, lacking a second copy of their genome. As such, they sequestered their germline as best as they could. Like root initials, these cells would very rarely divide, only when needed for branching or for creating sexual organs. Spores were also improved. An endosperm-like layer was added to provide the spores with food for early development. This was an intense version of matotrophy. Elaters were also added to allow the spores to fly further and better reproduce. The advent of lignin proved invaluable to some gametophytes who were able to grow taller and more robust than had ever been possible before. Improved vasculature, such as thinner xylem in the leaves and thicker xylem in the stem, allowed more water to be brought a farther distance. Thus, these gametophytes could avoid shading from their neighbors. Some gametophytes developed into epiphytes, taking advantage of other gametophytes' structure and sometimes even nutrients, saving them the trouble of having to build lignin bodies. The pinnacle of gametophyte evolution came in the advent of the angiosperm. Finding that insects were attracted to pollination droplets, there was an incentive to create sweet and colorful rewards, in short, flowers. Fusing archegonia and antheridium onto the same body, pollination could occur as insects moved from one plant to another. Thus, a complete and almost modern gametophytic plant world was able to evolve.